Okay, so we have covered the last of the material for Unit 2, okay, which is biology, okay, or life science. So we gotta start looking at what's gonna be on our unit exam. Okay, your unit exam will consist of 26 multiple choice questions and 9 written response. Okay. And obviously that will be written on Monday. Okay. Should you want to start early and get a little extra time to write, I will allow that. You will just have to find a way here to start early. If you normally take the bus, you got to find a ride. Okay, but I will let you start at 8:15, which will give you an extra 20 minutes, okay, to write. I don't think you'll need it, but whatever. If you can have it, makes you feel better to have a little extra time. All right, so key concepts in this unit. Okay, we started out with the cell theory. Okay, now are there some important people in the cell theory? Yes. Okay, do you need to know them all? Yes, there's only 4. Okay, so you just got to know Hooke, Leeuwenhoek, Schleiden, and Schwann, and you're done. Okay, it's not like the models of the atom where there was like a half a dozen or 8 of them. Okay, you don't have to worry about that. Okay, it's just these 4 people. Okay, and yes, you do need to know what they did, but only to recognize what they did. Okay, there's not going to be write a paragraph about the contributions of Anton von Leeuwenhoek. Okay, no. Okay, it's going to be stuff like, who was the first person to view cells, or who developed the cell theory, or something like that. Okay, simply recall kind of stuff. All right, so we got to remember that Hooke is the guy who initially discovered cells. Okay, and when he was looking at them, in a slice of what? Cork. Right. So the cells he looked at were dead. Okay, Leeuwenhoek was the first person to view actual living cells. Okay, and then after that came Schleiden and Schwann. Okay, who obviously have something to contribute there in terms of they developed the cell theory. Okay. Now, how important is the cell theory? It's pretty important. Okay, could there be a multiple choice question about the cell theory? Sure. Okay, there's 3 points to the cell theory. It sets up pretty well for a question like which of the following is not part of the cell theory, and you could have something like that. But more important than that is the second point of the cell theory. Okay, our cell anatomy labs were based entirely around this idea that cells carry out basic functions. By the way, when I'm done this, I'm going to go over your cell anatomy labs as well. Okay. But the second point of the cell theory about cells carrying out the basic functions of an organism is something that you can easily work into several of the written response items. Okay, when you're talking about cell transport, you can always work in the cell theory a little bit. Cells have to carry out the basic functions. Transport is one of those. Okay, or something along those lines. But it's a good idea, and there's lots of opportunities, okay, to work in that point of the cell theory into a few of your written response answers. David, question.
What does the MC decide?
Multiple choice. MC stands for multiple choice. WR stands for written response. Okay, so yeah, you could definitely work in that into a couple of your answers, and that kind of gives your answers a little bit of beef. Okay, just hey, I remember this thing, and I think it applies here. That's better than just saying, here's what I remember about this thing. Okay, you're saying I can incorporate other ideas into my answer, and that's a higher quality answer. Okay, so that's what you need to know for cell theory. Okay, and then after that, we talked about size estimation and microscopy. It is important for you to know how to estimate the size of an object, as I have put a question in the multiple choice about that. Okay, show you a picture of something I looked at under the microscope, and I drew a picture around it, drew a circle around it. Okay, and I'm going to ask you to estimate its size based on how much of the circle it takes up. Okay, so make sure that you remember how to do that. Okay, could there also be a couple of multiple choice questions about microscopy techniques? Like, how to use the microscope. Okay, there certainly could be. okay, you know, like how you carry it, or what lens you start on, or, you know, things like that, which one gives you the greatest field of view or the highest magnification, or, you know, things like that. Common sense kind of stuff, but it's still worth going over, okay? Lesson 2 there in your notes, or sorry, yeah, lesson 2, okay, where you've got some of that stuff. Okay? All right, and then this was one of the lessons that you watched while Mr. Van Erb was here, okay, because I was out sick, okay, but the modes of nutrition, that's lesson 4, okay, knowing chemo heterotrophs and photoautotrophs, okay, those are 2 terms that you should know, and in fact, those 2 terms cost a lot of people a mark in the analysis part of the cell anatomy lab, okay, as they were terms I was looking for in the analysis, okay? So, what does a chemo heterotroph have to do? It has to consume other organic material, usually other organisms, either living or dead, okay, for its energy. Whereas a photoautotrough can, yeah, it can synthesize them itself using photosynthesis. Okay. All right. Cell structure. There's a diagram of a cell on the test. You've got to be able to tell me what the parts are and what they do. Okay, not every part. I've only picked 5. 6. Okay, I picked 6 parts of the cell, so not the whole thing, and you don't know whether it's plant or animal. Okay, so you have to know both. That's not such a big deal, as really there's only about 4 or 5 structures that are common to both anyway, okay? But you need to know all the parts and what they do, okay? Because like I say, there'll be a diagram of a cell, and say, give me the name and the function of 1, 2, 3, 4, 5, and 6. Okay, and that's in the written. Now, there's also some stuff in the multiple choice about parts of the cell and what they do, so you also need to know that. Okay, and these would be more in the multiple choice, more application type questions, like you know, a cell in your body has to do this job, so its cells would be specialized to have more of what organelle, you know, mitochondria or cytoskeleton or, you know, something like that. So you really have to know what each organelle does, okay, to answer a question like that. Okay, and then, okay, cell structure, so that's labeling, okay, knowing compartmentalization, okay, that was a big word. Actually, it was one of the first things in lesson 5, which was another one of the lessons you did while I was away, okay. So compartmentalization is what makes a eukaryote different from a prokaryote. Okay, we're a eukaryote. Okay, our cells have organelles, a nucleus, a mitochondria, blah blah blah. Okay, prokaryotic cells don't have those. Okay, prokaryotic cells have no organelles. Okay, they simply have regions of the cell where stuff happens. Right, which is why prokaryotes don't get very large, and why there are no multicellular prokaryotic organisms. Okay, you can only be multicellular if you can specialize, and to specialize, you have to have different organelles. So that's one of the things that was in lesson 5. Make sure you go over that. Okay, there's a few questions in both the multiple choice and a way you can work it into a written response answer. Okay, so it's a topic that you should definitely review a little bit there. Okay, if there's questions as we go along, guys, don't be afraid to ask here. Okay, 5th thing, cell transport. Okay, so compartmentalization, like I say, I've got it written there twice, but cell transport, okay, what are the big 3 methods of cell transport?
Endocytosis.
Diffusion, osmosis, active transport. You need to know those 3 processes in detail. Okay, in detail, you must know those 3 processes. They are the second half of lesson 5. Okay, now diffusion, remember, is used to transport materials within the cell. Okay, from place to place within the cell. It's passive, so it requires no energy. Stuff moves from high concentration to low concentration, or down the gradient. Okay, It's not targeted and doesn't work well over large distances because it's not targeted and it's slow. Okay? What is osmosis transport? Water. Water only. Okay? And where does it transport it?
A high area of salt, like concentration of salt.
Right, it's gonna move, it's gonna make water move, and here's the key part, across a membrane to balance salt concentrations. Okay, so the water will move to the area where salt is higher in concentration. Okay, it'll move to that side of the membrane. It's the key part of osmosis. Okay, it only transports water and it moves it across a membrane to balance salt concentrations. If you get a question about osmosis on your unit exam, that's 3 marks. Okay, it transports only water across a membrane to balance salt concentrations. Okay, that's 3 marks. That's explaining to me I know what osmosis is. Okay, then you probably have a couple of marks about why that process is occurring or how it's applied in that situation. Okay. All right, and the third one is active transport. Active transport is different than the other two in what key way? Yeah, it's the only one that requires energy. Osmosis and diffusion are passive. Okay, the cell can't stop them. Okay, they simply go. But active transport requires the cell to invest energy. Okay, and it does that in order to pump materials against the gradient or up the gradient. Okay, that is, it's moving them from areas of low concentration to high concentration, the opposite of what the other two are doing. Okay, and that's also movement across the membrane. Okay, the key point. Okay, key points with active transport are uses energy. Okay, up the gradient or against the gradient, and what part of the membrane are they using? Those special little things embedded in the membrane. They change shape when the energy is applied to them. Nope.
Not like guard cells?
Nope. Transport proteins. Okay, so we better look over that one. There's little transport proteins embedded in the membrane. They act like a gate. Okay, when they're closed, okay, they would look like this. Okay, and then when energy is applied to them, they change shape and they essentially rotate like this, which opens them up, and then material can be pumped across. Okay, but it requires energy to change the shape of those proteins, and as they change shape back and forth, that's what creates the pumping action that draws material through. Okay, so it involves those transport proteins, and that's a key point. All right, so we got diffusion, we got osmosis, okay, we got active transport, carrier or transport proteins. Okay, we've even got it written here that that's an important thing to remember for that. Okay, diffusion, however, is the most important because it's the one that gets used most often by cells. Okay, it also relates to that cell size activity that we did. Okay, with the cubes, we were comparing the cubes that were imaginary cells of different sizes. Okay, remember that was to show that as a cell gets larger, the volume through which it must use diffusion increases very quickly, and diffusion very quickly becomes ineffective over those larger volumes and distances. Okay. Okay, multicellularity. Okay, so that is, okay, why is it an advantage? Okay, and to be multicellular, you must have this. Okay, this is the key here. Okay, all multicellular organisms have specialization. The example we went over was what?
Leaves.
The layers of the leaf. Okay, you should definitely know the different parts of the leaf and know them for this reason or in this context. Okay, know what each different part of the leaf does, because that's an example of specialized cells doing a certain job, working together for the benefit of the whole organism. Okay, so we looked at how the epidermis has one job, it secretes the waxy cuticle. Okay, we looked at how the palisade layer's job is to carry out photosynthesis. Those cells are packed in really tight. Okay, they have a definite shape and they're full of chloroplasts. Okay, because they have one job, carry out lots and lots of photosynthesis. Okay? The spongy layer underneath still has lots of chloroplasts, but the cell's shape is way different, and that's in order to create spaces, okay, within that layer where water and gases can be exchanged. Okay, it's access to the stomates, okay, where that stuff can be exchanged, where water can evaporate, carbon dioxide and oxygen can switch places. Okay, so look at those in that context. What does each layer do, or what does each type of cell do? How is it specialized to do that? Okay, there's going to be a question that asks you about that. Okay, you need to know that as an example of a multicellular organism. Okay, what are the advantages of multicellularity? Okay, well, the big original advantage was it allowed organisms to get bigger. Yeah, okay, bigger was better, okay, but as a single cell, there was certainly an upper limit on your size. Okay, and that was because of the surface area to volume ratio problem we talked about in that cell size activity. Okay, so the advantage of multicellularity, yes, it allowed organisms to get bigger. Further to that, it allowed them to become more complex. Okay, and that allowed them to fill different niches that were available and things like that. Okay, so multicellularity was a huge advantage for those reasons. Okay, examples of specialization implants, that we just talked about this, okay, that's the layers of the leaf. Okay, so make sure you know that one, it's kind of been talked about twice now. And then, okay, transport implants. Okay, you need to know the tissues of the plant responsible for transport, right? So that would be what, two things? Xylem and phloem, okay? Know what each one transports and how that is accomplished. Okay? Especially water, right? Because we did a lab on that. Just finished it the other day, okay? Make sure you are able to explain how a plant transports water. Something else you're going to need to explain. And it's not something we went over overtly, okay? But it is something you're going to have to kind of apply your knowledge to. Thinking about how a plant functions, you're going to be asked to explain how a plant can control and conserve water loss. Okay, so you need to think about what structures on a plant help a plant to regulate or prevent the loss of water, including any adaptations that it might show or growth patterns that it might show in order to make that happen. Okay, so that's kind of apply your knowledge of plant growth and development as well as transport and structure, okay, into one question. Okay, so like we said, be able to explain that in detail and apply your knowledge. Okay, parts of the plant, so transport in plants has to do with phloem and xylem, but they're also parts of the plant. Okay, the leaf, the stem, the roots, what are their jobs? Okay, that was in your quiz yesterday, right? That kind of stuff is going to be tested mostly in the multiple choice because it's mostly recall. Okay, so phloem, xylem, we already talked about this. Where are they? Okay, what forces do they use? Okay, stuff like that. Okay, and then plant growth and development and adaptation. Right, this is where you're going to need to know your plant hormones as well as the tropisms and how the tropisms are, how they're accomplished. Okay, and I think I told you this the day we were going over tropisms, but the way I'm going to test that is I'm going to put a picture of a plant, okay, on the test, and I'm going to explain the situation to you, and you are going to have to tell me what tropism is mainly responsible and how it happened. Okay, that means explaining in detail how that tropism occurs. Basically how all tropisms occur. Okay, the causes are different, but the mechanism is the same. What's the mechanism for a tropism?
Certain, like the cells on certain sides of the stem get expanded.
Okay.
And the plants.
Right. One side of the plant grows faster than the other. Differential rate of growth was the way we stated it in the lesson, okay? And then you have to explain, all right, how does that happen? Well, okay, are there plant hormones that make, you know, part of it grow faster? How would that work? Okay, be able to explain that, okay? Okay, so hormones, tropisms, okay? How that's helping a plant adapt to its environment. Okay, and then lastly, factors affecting plant growth, biotic and abiotic components. We talked about that. Okay, that would be all multiple choice. Okay, but I mean there could be a way to work that into a couple of the written response questions as well. Okay. How do you go right on this exam? Okay, what are some things you need to do to be successful on this test? And I would say the thing you need to do best on this exam is explain everything well. Okay, what I mean by that is this. If you're doing that question I was telling you about the tropisms, okay, and it says something like, you know, explain what's happening to this tree and what's caused it to happen and how that process occurs. And all you write is phototropism grows towards the light. And it's a 5-mark question. How many marks are you going to get for that? Maybe 2. Yeah, okay. But if you said, this is caused by phototropism, which is a growth response to the location of light, and is, in order to accomplish this type of growth, the plant needs to make cells on one side grow faster than the other. In order to do this, auxins are secreted in larger amounts to that side of the plant, causing those cells to elongate, okay, making the plant curve, directing its growth in the direction of the light. That would be a 5-mark answer. But the mistake I see people make most often on this exam is that they don't put in enough detail. Okay? And even though I'm sure they know more than they wrote, I can only mark what you write. Okay? So if you don't write all the stuff you know, you're not going to get marks for it. Okay? So that would be my biggest suggestion for you. Okay? If you think it's important, write it down. Okay? Now, there's a danger to that. Be careful you do not contradict yourself, because I've seen people do that. Okay? They write too much, and they actually start going back on what they said. Okay? So they're talking about how this plant is using only phototropism and growing towards the light, and then they say, yeah, and it could also be geotropism. And now suddenly they're going to lose marks because they don't know which one it is. Okay? And they've gone back on what they said, and now it's unclear. Now it seems like I just wrote everything I knew down in the hopes that some of it was right. Okay, well that's great. Some of it might be right, but if it contradicts itself, then it says I don't know what I'm talking about. Okay, and there's that danger as well. Kind of pick one and go with it. Okay. All right, so we said 26 multiple choice and 9 written response. Okay, and the types of things that are in there. Okay, there's some, like I said, microscopy. So I'm talking about the multiple choice here, guys. Microscopy, cell structure, cell transport, cell structure and function, cell structure and function, cell structure and function, functioning of a multicellular organism, cell transport. Oh, sorry, with cell transport. So we talked about the big 3. Were there 3 other methods by which material can move into or out of the cell? Okay, this is where you talked about phagocytosis, Pinocytosis, and receptor-mediated endocytosis. Okay, all of those involve changes of shape of the membrane, so they're not like the big 3. Okay, they're ways that a cell either engulfs to intake material or expels material. Okay, those, again, they all involve changes in shape of the membrane, okay, whereas the other three don't. Can we go over those? Yeah, I can go over those in a minute. Okay. So then there's some cell theory stuff. Plant structure and function, plant structure and function, plant structure and function, plant growth and development, 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 modes of nutrition, multicellularity, microscopy, okay, that's what's in there. All right, and then in your written response, you've got plant growth and development, cell structure, so that's your cell diagram. Okay. By the way, that cell diagram is a 15-mark question. Okay. And then multicellular organisms, plant transport, cell transport, plant growth and development, and cell transport. Okay, so those are the topics covered. Okay. In your 26 multiple choice and 9 written response, the entire test is out of 78, which is 3 marks less than the chemistry exam. So they're almost exactly the same. Now, difference obviously with biology is you've got a lot more writing and explaining than you did in chemistry, which was all demonstrate your skills. Okay, so this test is going to be a bit different when you write it in that regard. Okay. All right, so the request was, can we go over those methods of cell transport? So we have a look at those, and then I'm going to go over your cell anatomy labs. Okay, so this was one of the lessons you guys went over with Mr. Van Erf. Okay, it was all different parts of the cell, and then this was the cell transport part. Okay, you talked about osmosis. This was the active transport part. Okay, carrier proteins, transport proteins, they open and close when the energy is applied to them. That's how active transport works. Okay, and then we were looking at exocytosis, which is movement out of the cell. That again involves a change in shape of the membrane. Okay, osmosis doesn't involve change in shape of the membrane, diffusion, nor active transport. Okay, these all involve either something being engulfed or expelled. Okay, a vesicle is either produced or ended in these processes. All right, so in exocytosis, okay, a vesicle produced by the Golgi goes to the membrane. Since it's made of the same material as the membrane, it simply, when it touches the membrane, it merges with it, and it simply expels whatever was inside. Okay, and then that vesicle just becomes part of the membrane. Right? And then later, that same part of the membrane could engulf something, and that piece will become a vesicle again, and it'll go and become a lysosome by merging with a peroxisome or something like that. Okay, so the membrane's always folding in and creating these vesicles and stuff like that. Okay, and the process is very much like, how many of you like have a dog? Okay, when you walk your dog, you have to take the little poop bag, right? Okay, very much like that. So when the dog poops, you put the bag around your hand and you pick up the poop and then you turn the bag inside out. Okay, that's kind of how the cell membrane works. Something touches the cell membrane and just kind of encloses around it, turns inside out, and yeah, then you got a vesicle with stuff inside. Okay, obviously the process is opposite. Cell getting rid of things like poop. Cells don't poop, but they get rid of waste. Okay, kind of a similar process to that. All right, now 3 ways that things get through the membrane by the membrane changing shape. Okay, the first type is phagocytosis, which is the cell engulfing a solid particle like food. Okay, so most of this time this process involves engulfing food, but if it's a white blood cell, it's also how the white blood cell would kill like invading microorganisms like bacteria and things like that. Okay, so all the cell does, okay, is it simply creates what are called pseudopods, basically extensions of the membrane. The membrane just changes shape and kind of morphs, okay, until it's giving this thing like a big hug. okay, and then it just merges on the other side of it, three-dimensionally, of course, okay, and it creates a bubble, and now that object is encased within the bubble. And with a white blood cell, like this diagram is showing, it's simply engulfed the bacteria, okay, peroxisome or lysosome will merge with that vesicle, and the digestive enzymes will kill the bacteria, okay, and that's how your, like leukocytes and things like that work. Okay, so phagocytosis engulfs a solid particle. Okay, Pinocytosis, same process, but engulfs only fluid. So if a cell is really dehydrated, this is a really quick way for it to rehydrate. Okay, rather than waiting for osmosis to passively move the material through, okay, it will simply gulp huge amounts of water. Well, not huge as a cell, but huge for a cell. Okay, it'll just start gulping it, right? And you can actually see that this is an electron microscope picture here, okay, on the right, of the cell actually engulfing simply fluid. Okay, this is pinocytosis. You can see one vesicle after another being formed here. Okay, so it's just engulfing the intracellular fluid. Okay, so that's pinocytosis. So phagocytosis, solid particles; pinocytosis, fluid. Okay, and then the last one is specific. It's called receptor-mediated endocytosis. So your cell membrane is covered with things called receptors or receptor proteins usually, right? These receptor proteins have a certain shape. They're kind of like the plugs on the back of your computer. Okay? Every single one of them is a different shape. Okay? Like you might have 6 USB ports, and then you've got your port for your monitor, a port for, you know, whatever else. Okay? It used to be worse because now everything is USB, but before, okay, a long time ago, everything had a different shape. And you had your keyboard one, you had your mouse one, they were all different. Okay? It's kind of like that, okay, in that all the receptors have a different shape. It's almost like they have to have their, you know, a different antenna for every signal they could receive, rather than having one antenna that can receive all signals. They have many antennas, each one for a separate signal. So when a material, like a hormone, a chemical messenger, okay, hits that receptor, the receptor then triggers a reaction within the membrane, causing it to do phagocytosis, okay, and it just engulfs that hormone, okay, because the hormone is a protein, and it's way too big to just go through the membrane, okay, it can't pass through that layer, so it has to be engulfed, okay, and it gets engulfed in a vesicle, okay, and then that vesicle, okay, can be taken to wherever it has to go within the cell, right? And you can actually see that this is an electron microscope picture of that happening, right? You can see all the little, the coat proteins or receptor proteins, okay, along the membrane here, okay, and these ones have all received their chemical messenger. That's kind of what the gray blurry stuff is. Probably the best example is right here, okay? It's almost, there's the receptor, okay, and the molecule sitting in it is this. Okay, kind of looks like that. It's received its chemical messenger, okay, and it's causing the membrane to envelop it. Okay, you can see that here. Here's all the messenger molecules, okay, like that. Okay, and you can see here, this is the membrane. Right, so this vesicle has almost been like pinched off. Okay, so it's almost completed. All right, so those are the 3 bits. That cover what you were looking for? Good.
So for each thing that the cell would need to take in, there's a receptor for that?
Yep.
And the thing, and the receptor needs to receive the thing that it's specialized for to do this?
Yes. Now, it wouldn't be necessarily for each specific thing, but maybe like a group, okay, of signals. So anything that's, you know, in the group of adrenal compounds would probably have just a type of receptor for adrenal type compounds, and ones that had to do with pancreatic, so glucagon or insulin would probably have similar shape. It would take a group of hormones. All right. Any other questions there, guys, or anything else you want me to cover again? No? Okay, then let's look at your cell anatomy.